Hello everyone, welcome to Biopatrika YouTube channel. Today we have with us Professor Aswin, who will be talking about his new book, Bacterial Genomes, Trees and Networks. But before we deep into this book, let's first hear about Dr. Ashwin.
I'm a member of the faculty at the National Center for Biological Sciences in Bangalore, part of the Tata Institute of Fundamental Research, an autonomous institution under the Government of India. My lab works on aspects of bacterial genomes and how bacteria adapt to their environments. And we typically use computational methods which make use of large volumes of publicly available genomic data on a variety of organisms to try and answer various questions on bacterial adaptation and evolution and how they regulate their physiology. So that's what we do now. In my education background, I did my bachelor's, a BTech in biotechnology in Anna University, Chennai. Finished about 20 years ago. And then I did my PhD in Cambridge at the European Bioinformatics Institute with Nicholas Kum and joined NCBS in late 2010. I've been here since.
Thanks a lot for your brief introduction. So let's dive into this particular book. So, what inspired you to write Bacterial Genome Trees and Network book? So, what was the vision or gap you tried to fill with this book?
Okay, um, so this is I started writing this book sometime in 2022, and just after we were trying to get back on our feet after COVID, and during the COVID period, we had actually finished a whole bunch of projects, and several students had finished their PhDs and I left, and I was trying to also do something different, the kinds of science that we are doing. And while I was thinking about that, I came across this review by Roberto Holt, it's um, a legendary microbiologist. And in this a book, he talked about the history of microbiology and how what he calls as unified microbiology, which is microbiology as a very broad science without having different silos, has over time uh, been divided into different sub things. For example, medical microbiology, which is interested only in microbiology from a disease point of view, and molecular microbiology, which is interested in molecular biology, but they're taking out the the ecology of bacteria from the equation, focusing on these molecules, and that now um, uh, the more um, microbiome type research, which tries to do more ecological microbiology, is probably trying to unify microbiology once more. So that was this perspective that he had written. And then I was thinking, okay, the kinds of questions that we are interested in, which is microbial um, adaptation to their environment, but trying to do so at a fairly molecular level by looking at evolution of things like regulatory systems and how regulatory networks function, how they are structured, how they evolve, and how genomes themselves change over time, probably supporting adaptation. It's also a way where we're trying to bring an ecological um, aspect to microbiology, but they're also taking a molecular angle. And then I also realized that uh, in colleges, um, when one is teaching genomics, looking, it's very very technical, um, and it, somewhere along the way, uh, the fundamental biology behind why we are doing genomics seems to get lost. So I thought, okay, now we're changing fields, and I also have time to write something new and also learn something new along the way. Thought, okay, why not write a second book? It's been close to a decade since I wrote my first. So that's where it started.
Yeah, that's good to know. So, what was the inspiration behind this book? So, the title mentions trees and networks, so which is again thought-provoking. So, how do you think these metaphors reflect bacterial genome evolution?
Right. So trees are pretty much the fundamental view of evolution, starting from Darwin and Ernst Haeckel's time, and because evolution is predominantly divergent, presumably from a single common ancestor, then it goes like from the base of a tree all the way to the leaves. And extant organisms are what we call as leaves. And as we go further in into looking at nodes, these are all ancestral nodes and so on. So that was a fairly obvious term to use for something on evolution and molecular evolution. And the networks idea has at least two interesting links to this book. One is that the final chapter talks about regulatory networks. While evolution does help in adaptation over certain time scales, within the uh, time period of a single cell before it starts to do, before it starts to reproduce and then genetic evolution can take place, physiological adjustment to changes in environments happen through some kinds of regulation. And this is typically at the transcription level, where gene expression states are change. Um, and these kinds of changes are orchestrated by what are called as regulatory networks. So, where you have a regulator regulating a target gene, the regulator becomes a source node, and the regulated target gene becomes a recipient node, and there is an edge drawn between the regulator and the regulated. And this forms a fairly large and complex network in many organisms. And so there's one, and that's something that we talk about. And the other network goes back to molecular evolution once more, though we have a tree-based representation of phylogeny. Um, the idea that of horizontal transfer, which is very prominent in bacteria, where bacterium acquires a gene not from its parent, as we all do, but from something that coexists, which may not be phylogenetically very related, which means that certain uh, genes, there maybe a very, very distant organism gets connected to some bacteria. So you have the tree on which you can draw a network of horizontal transfers coming from um, one end of the tree to the other. So the network comes from those two angles, one from horizontal transfer and from physiological regulatory networks.
I think that's an interesting perspective and the way how we visualize these networks and evolution. So your book is again blend of historical milestones with modern science on bacteria or microorganisms. So why do you think it is important to bring in this historical perspective when you were writing this book?
The motivation for this is come from the extraordinarily rapid growth of technology in molecular biology, especially sequencing technologies, and the way we can use these sequencing technologies to do a lot of things. It's not just to get a DNA sequence, but we also get quantitative information. We can even get information on the structure of the chromosome just from these sequencing technologies. So this is changing very, very, very rapidly. And so I mentioned earlier, and these some of these technologies and techniques are have been the focus of a lot of our education. which is good. So because one gets awareness about how to do things, but at the same time, the speed at which these things are changing uh, means that probably what I'm learning in college today, by the time I finish my course, is probably an outdated technology replaced by something else. And what doesn't change or changes slowly are the concepts. And when we look when we go back in history and to see from the beginnings of microbiology 300 years ago, or at a faster pace 150 years ago, a lot of things have taken a long time to come through. It took, I don't know, 270 years, something close to 300 years for us to even recognize what bacteria are or what their place is in the tree of life. Of course, improvement in technologies will increase the pace at which we find things, but the rate limiting step has always been finding the right question and finding the right approach for that. And I believe that to try and understand how this has played out over time gives some perspective on the importance of being thoughtful about what we are doing, and that the instant gratification usually uh, doesn't uh, create a significant impact. Uh, so to bring that perspective, okay, it's probably good to say, okay, I know this is a concept we talk about, a lot of genomes and so on, but this has been built over 150 years of thought and experimentation by a lot of people from different parts of the world. So that's why I thought a historical perspective would be useful. Um,
Yeah, I think it makes sense now to bring in that historical perspective so that we understand exactly how it started and from where it is like coming to the current scenario. So,
And it was also a good learning experience for me to be out of my comfort zone there. Some of the literature from, I don't know, 50, 60 years ago, I don't even know what the terms mean anymore. We never studied them. So it was a challenge.
so coming to another important question. So, how do you think this book is different from regular genomic textbooks, which we study in universities or maybe colleges?
I think the biggest difference is that this is more concept focused. Um, whereas most genomic texts and genomic curricula also often tend to be techniques and technology, and yeah and uh, technologies change quite rapidly, well, concepts change far more slowly, and I believe that learning the concepts is far more fundamental, and doing that well is important, and that is probably being given the short shrift in most institutions, obviously with exceptions.
So, what do you hope an undergraduate or early career researchers will take away from this book, and what should be the mindset when someone is reading this book?
So I I hope that uh, they would um, first recognize that uh, basic sciences, asking fundamental questions in bacterial physiology, is interesting, and studying bacteria not all about antimicrobial resistance or disease and that sort of thing. That there is more to this than that, and also that a lot of things in science is mostly about the process than the outcome. As Virat Kohli would say, process. It's not it's the same thing. Um, uh, Getting the process right and having a deep understanding of uh, whatever we are studying is at least as important as finding a solution uh, uh, to a present a problem. And I I hope that the book will give, generate some interest in that sort of basic questions and interest in trying to uh, answer those. That's the goal.
Yes. So, while writing books, what were the challenges to like keep it simple as well as like more informative?
Yeah, it's I think to try to keep the jargon to a minimum, and in fact it wasn't too difficult at this point, because at this point my own ability to focus on the extreme detail has declined so rapidly over the last 5 years that uh, I'm also becoming more superficial in how I look at science. And then I realize it's not such a bad thing at the end of the day. Sometimes there is a phase in life where that detail is important. But uh, before that, which is when you're in an undergrad learning something, and maybe after that phase is over, it's fine not to focus on the detail, and be a bit more philosophical about things. So uh, at this point in time, it was not the hardest thing for me to do. I would have struggled 10 years ago to do that, but now, yeah, I think I'm happy being more like an undergrad than the detailed oriented PhD student.
So, talking about bacteria and bacterial evolution, so we all study about like antimicrobial resistance and many other diseases arising from resistant bacteria. So, whether this book touches upon this modern day issues or it just talks about like the basic bacterial evolution.
It does. So it starts off. It tries to get a concept across, and actually, antimicrobial resistance is one example of uh, bacterial evolution, and it is we. I do talk about this in at least two different areas. One from the historic uh, perspective. And to me, it was a surprise to learn that um, uh, we're known about the resistance from 1920s. And it's though it really in practice and in terms of popular press, it really went out of the charts in only the 1990s. But we've always known about them from 1920s, and they've always been warning bells, but we've just ignored them. So that's from the historic perspective. And again, when talking about mechanisms of evolution, horizontal transfer, mutations, and so on, we bring back some examples of antimicrobial resistance. So it's there. It's there.
Yeah, thanks for I think touching upon the different aspects of like bacterial evolution and the diseases linked to it. So, finally, what message would you like to give to young students who might be thinking of stepping into field of microbiology or genomics?
I would ask them to slow down, and uh, keep in while finding applications and solving problems is very, very important. And many people do that and do that well. And there is also space to grow knowledge about this field. There's uh, one of these books, which is an essay. Or there is this very famous essay on called the usefulness of useless knowledge. Um, and there is a Princeton University Press edition of that essay with an accompanying more modern article. And there the author talks about how knowledge is pretty much the only resource that grows the more you use it. It is probably the only Akshay Patra thing that we that we know of, and growth in knowledge is usually from basic sciences, and the basic sciences move forward slowly, but a good proportion of them do have major impact on society at some point, if not immediately, no, at some point, and that plus imagination um, are probably the most important things, and there is space in this world for that. And if a good proportion of young people um recognize that and go for the basic research, um, not just abroad, but hopefully also in India, I think we'll be fine. And that's the idea that I wanted to get across.
Thanks a lot, Professor Aswin, for joining us today and giving us what to expect in your new book on bacterial genomics. And I'll definitely leave the link in description for people who might be looking forward to read this book and know more about like historical perspective as well as how it is linked to today's issues related to bacteria and its evolution. So thanks once again for joining us. And people who might want to learn more about this will, they can definitely reach out to you. We'll leave your email in description.
Yes. Thanks, Virendra.